Hello, this is Diana Cervantes and Katie Countryman's video on mitosis. Mitosis is a phase in the cell cycle when it is time to divide and make two new nuclei. The cell spends 90% of its time in interphase, where it is growing and preparing for cell division. In the G1 phase, that's where the cell begins to grow in size, so that way when it goes through the S phase, the chromatin, which are long stringy fibers, begin to create copies of itself. In the G2 phase, the cell grows again, and the single centrosomes, which are located outside the nucleus, these chocolate chips, along with the centrioles and the microtubules, which are the noodles. When the cell goes through mitosis, it goes through 5 subphases: prophase, prometaphase, metaphase, anaphase, telophase, and then the next phase the cell goes through is cytokinesis. This is when the cytoplasm of the cell separates and the results are 2 new cells. Once the cell goes through all its stages in interphase, the cell is ready to go through the first subphase of mitosis, prophase. Inside the nucleus, the chromatin begin to condense into thick chromosomes. The copies made during the S phase of the chromosomes are called sister chromatids, which are connected at the waist, called the centromere. While the chromatins are condensing, the nuclei begins to disappear, and in the cytoplasm, the microtubules begin to expand from the centrosomes, and the mitotic spindle begins to form. When the 2 centrosomes move to the opposite sides of the nucleus to expand the microtubules and the mitotic spindle fibers between them. After prophase, it goes right into prometaphase. In this subphase, the nuclear envelope disappears completely, which allows the mitotic spindle fibers to enter and interact with the chromosome. At this time, the chromosomes are fully compacted and create new structures called kinetochores, which are located in the centromere area. This is where the microtubule fibers move towards the center of the cell and attach to the chromosomes. The other microtubule fibers, non-kinetochores, push against each other and stretches the cell. The next phase is metaphase. This stage is the longest of mitosis. It is where the chromosomes line up along the metaphase plate, which is the center of the cell. The spaghetti noodles, or the spindle fibers, attach to the chromosome kinetochore, or the blue sprinkles. The microtubules that are not attached to the kinetochore attach to the metaphase plate and are called spindle. After metaphase is anaphase. Anaphase is the shortest phase of mitosis. The 2 sister chromatids begin to part separate ways, and each chromatid becomes a full chromosome. Then the chromosomes begin to move towards the opposite ends of the cell, where the centromeres are. The centromeres are first to be pulled in because the microtubules begin to attach to the centromere region. Telophase is the last stage before the cell fully splits into 2 nuclei. The middle of the 2 cells splitting is called the cleavage furrow, but that only happens in animal cells. The nuclear envelope begins to reappear in fragments. The nuclear points of the parent cell's nuclear envelope in the other part of the endomembrane system. The nucleolus begins to form again in each nuclei. In late telophase, cytokinesis begins. Cytokinesis is the division of the cytoplasm so there are 2 new daughter cells at the end of telophase.